Hello and welcome to this tutorial on exploring biological sequence data and file formats. In this video, we will cover the essential aspects of bioinformatics resources such as UniProt and NCBI. This exercise aims to familiarize you with key bioinformatics tools and develop your skills in retrieving and analyzing biological sequence data, interpreting protein functions, and understanding the significance of various protein modifications and interactions. Let's begin by exploring the UniProt entry for the protein Tyrosine-protein kinase ABL1 with the UniProt ID P00519. Our aim here is to explore and understand the detailed information available for the protein Tyrosine-protein kinase ABL1 in UniProt. This includes examining the protein's function, structural interactions, subcellular localization, disease association, and sequence information. First, we need to access the UniProt entry. So go to the UniProt website. The official web address for reaching UniProt is www.uniprot.org. Enter the protein ID P00519 in the search bar and click on this and click this search button. Once you have accessed the entry, let's navigate through the different sections to understand the detailed information available. The first section at the beginning is the overview section. Here, review the basic information including the protein's name, gene name, and organism. The next section is the function section. Read through the detailed description of the protein's function, including its role in enzymatic activity and cellular processes. The next section is the names and taxonomy section. So note the recommended name, synonyms, and taxonomic details. The next section is subcellular location. Observe where the protein is localized within the cell from this section. The next section is disease and variant section. In this section, examine the information on associated diseases and documented variants. The next section is PTM processing section. Review the post-translational modifications and processing information from this section. The next section is expression section. In this section, look at the expression patterns across different tissues. The next section is interaction section. Check the protein-protein interactions listed. The next section is structure section. In this section, go through the structural information provided for protein of our interest. The next section is family and domain section. In this section, identify the family and domains associated with the protein of our interest. The next section is sequence and isoform section. In this section, just go through the protein sequence of our interest and any isoforms present for the protein of our interest. Also, display the protein sequence in FASTA format. For displaying the protein sequence in FASTA format, click this download option. So this is the protein sequence, ABL protein sequence in its FASTA format. So the FASTA format always starts with the greater than symbol. The last section is the similar protein section. In this section, just explore proteins that are similar to ABL1. So we have explored all the sections which are given in the UniProt. After completing these steps, make sure to answer the given question to reinforce your understanding. Next, let's investigate the post-translational modifications of the high-affinity nerve growth factor receptor with the UniProt ID P53355. Our aim here is to investigate the post-translational modifications of the high-affinity nerve growth factor receptor and identify the specific residues affected by this modification. First, we need to access the UniProt entry for 53355. So go to the UniProt website, which is www.uniprot.org. Enter the UniProt ID P53355 in the search bar. So click on the search button. So let's navigate through the entry to focus on the post-translational modifications. But before that, I highly recommend you to go through this overview section, review the basic information, protein name, gene name, organism and all. Then go to the PTM processing section. Just focus on the details provided for post-translational modifications. Make note of all the details. After completing these steps, make sure to answer the given questions to reinforce your understanding. Next, we will retrieve and analyze the FASTA sequences of GABA transaminase from 4 different species: human, mouse, pig and chicken. Our aim here is to retrieve and analyze the FASTA sequence of GABA transaminase from 4 different species. So we have to retrieve this sequence from NCBI, not from UniProt. So first we need to access the NCBI database. So for open the NCBI website, whose official web address is www.ncbi.nlm.nih.gov. So in this we have different databases. We have to choose the protein database because we are going to search for the protein sequence of GABA transaminase. So we have to do this search specific for 4 different organisms: Homo sapien, human, mouse, pig and chicken. So the scientific name of human is Homo sapien, the scientific name of mouse is Mus musculus, the scientific name of pig is Sus scrofa, and the scientific name of chicken is Gallus gallus. So first I am going to demonstrate how to do this search for human. So we are going to search for GABA transaminase sequences of human. So for doing that, we will do the search for GABA transaminase and click on this search button. Now, to make this search specific for Homo sapien, we'll go to this advanced option. We'll type again GABA transaminase. So here this is all field. So I am going to choose another field, that is organism. So in the organism, I am going to type Homo sapien and do the search now. So the advantage of this search is it will retrieve only the gamma transaminase sequences from Homo sapien. So here you can see there are 22 entries which are related to GABA transaminase protein of human. So now we have to select the relevant GABA transaminase entry from the search result. So I am going to choose this one, which is mitochondrial isoform 1 precursor protein. Click on this. Here you can find the details of the sequence. Now we have to display this sequence in FASTA format. So for doing that, click this FASTA button. So this is the sequence in FASTA format. The GABA transaminase sequence, mitochondrial isoform 1 precursor protein sequence in FASTA format. Repeat these steps for the other 3 organisms, mouse, pig and chicken. After completing these steps, make sure to answer the given questions to reinforce your understanding. Now, let's move on to some self-evaluation tasks given in the manual. The task 1 is exploring GenBank. They asked 2 theoretical questions. The first question is, explain the significance of GenBank in the context of molecular biology and genetic research. The second question is, what are the primary functions and benefits of using GenBank for researchers? The second task is retrieving the coding sequence of human P53 from GenBank. So for this task, you have to go to NCBI and do the search for human P53 against the GenBank database. The questions what they have asked is, how can you retrieve the coding sequence CDS of the human P53 gene from the GenBank database? Detail the steps involved. The second question is, provide the accession ID for the human P53 gene and the total length of the coding sequence. The third task is fetching the protein sequence of miraculin from UniProt. So you should go to UniProt and search for miraculin. So explain the process for retrieving the protein sequence of miraculin from the UniProt database. Provide the UniProt accession number for miraculin and its sequence in FASTA format. For this task 3, you have to do the search for all the 4 species, that is Homo sapien, Mus musculus, Sus scrofa and Gallus gallus. That's human, mouse, pig, chicken. So you have to do the search of miraculin for all the 4 species. By completing this exercise in this module, you have gained practical experience in navigating major bioinformatics databases like UniProt and GenBank. You have learned how to effectively retrieve and analyze biological sequences, interpret various bioinformatics data formats, and understand the functional roles and modifications of proteins. These skills are vital for anyone pursuing a career in bioinformatics, molecular biology, or related fields. Thank you for watching. Don't forget to like, subscribe, and leave a comment if you have any question. Stay tuned for more tutorials.